Introduction. Welcome to today's lecture on the intricacies of the cell cycle and the molecular mechanisms that govern its progression. In this session, we will delve into the roles of various factors such as cyclin-dependent kinases, CDKs, cyclins, and checkpoints in ensuring proper cell cycle control. Additionally, we will explore the consequences of defective checkpoint functions and their implications in diseases such as cancer. So, let's begin our journey into the fascinating world of cell cycle regulation. During the G1 phase, CDKs and cyclins work together to regulate cell cycle progression. CDKs are protein kinases that phosphorylate key substrates, and cyclins bind to CDKs, activating them. This CDK-cyclin complex then phosphorylates target proteins, promoting entry into the S phase. In response to DNA damage, the tumor suppressor protein p53 is activated to initiate DNA repair or induce cell death. Its regulation through post-translational modifications is complex and crucial in preventing cancer development. Further understanding may aid targeted cancer treatment development. The G1 checkpoint ensures that the cell is ready for DNA synthesis. If DNA damage is found, the cell stops dividing to repair it. A protein called p53 is important in controlling this checkpoint, activating genes that pause the cell cycle. Repair enzymes are also recruited to fix any damage. These checkpoints are crucial for maintaining genomic stability and preventing the occurrence of genetic abnormalities. Additionally, the G2 checkpoint ensures that all DNA was replicated accurately during the S phase and that any remaining DNA damage is repaired before the cell enters mitosis. Ultimately, these replication fork checkpoints play a vital role in maintaining the integrity of the cell's genetic material and preventing the development of diseases such as cancer. These repair pathways involve the recognition and removal of damaged bases or removal of the entire damaged DNA strand. The repaired DNA is then ligated, or joined back together, to restore the integrity of the genome. Failure to repair DNA damage during S phase can lead to mutations and potentially contribute to cancer development. The S phase checkpoint is intricately regulated by signaling pathways that ensure proper DNA replication and prevent the propagation of damaged DNA. ATM and ATR, mentioned earlier, are key players in initiating these pathways. In response to DNA damage, ATM and ATR activate downstream effector kinases, which in turn phosphorylate target proteins involved in DNA repair, cell cycle arrest, and apoptosis. Additionally, signaling through the checkpoint kinase CHK1 and CHK2 further enhances genome surveillance by amplifying the cellular response to DNA damage. At the G2 checkpoint, the cell ensures that DNA replication is completed and DNA damage is absent before proceeding to mitosis. The CDK1/cyclin-B complex is activated and regulated, facilitating the phosphorylation of key substrates that promote mitotic entry. ATM and ATR are protein kinases that respond to different types of DNA damage. When activated, they initiate signaling pathways to repair DNA, arrest the cell cycle, or induce cell death, ensuring the integrity of the genome. To ensure proper DNA repair before entering mitosis, cells have mechanisms in place to delay cell cycle progression in G2. One such mechanism involves the inhibitory phosphorylation of the CDK1/cyclin-B complex. Phosphorylation of CDK1 by kinases such as WEE1 and MIT1 inhibits its activity, preventing premature entry into mitosis. Conversely, the activation of kinases such as CDC25 by ATM and ATR removes inhibitory phosphorylations, allowing the CDK1/cyclin-B complex to become active and drive the cell into mitosis once DNA damage is repaired. The spindle assembly checkpoint, SAC, is a surveillance mechanism that ensures accurate chromosome segregation during mitosis. It monitors the attachment of kinetochores, protein structures that connect chromosomes to microtubules involved in chromosome movement. If kinetochores are not properly attached to microtubules, the SAC remains active, inhibiting the anaphase promoting complex, APC/C, and preventing the separation of sister chromatids. This checkpoint gives cells more time to correct any attachment errors, ensuring the accurate division of genetic material. The M-phase checkpoint control ensures proper entry into anaphase and the subsequent completion of mitosis. Several molecular mechanisms and signaling pathways are involved in coordinating this checkpoint. The APC/C, mentioned earlier, is a crucial component of M-phase checkpoint control. It targets specific proteins, including securin and cyclin-B, for degradation, allowing for sister chromatid separation and exit from mitosis. Additionally, regulatory kinases, such as the polo-like kinases and CDK1, contribute to the coordination of different events during mitosis. Defects in the M checkpoint can lead to chromosomal instability and the accumulation of aneuploid cells, which have abnormal chromosome numbers. Aneuploidy is a frequent characteristic of cancer cells and is associated with tumor development and progression. Chromosomal instability arises due to the improper segregation of chromosomes during mitosis when the M checkpoint is compromised. This can result in the gain or loss of whole chromosomes or chromosomal segments, disrupting gene dosage and leading to genomic instability. In summary, the cell cycle is tightly controlled to maintain genetic integrity. Its understanding is crucial for disease management and scientific progress. Thank you for joining me on this journey of discovery.